Thank you, Merlin, for that introduction. I get to get away from my job as an AV guy. Of course, the computer just crashed, so we're on our backup computer, so hopefully we're good. But Merlin thankfully talked long enough to sort of cover that. So hopefully what we're going to wrap up here today is really looking at copper. And I don't know that I have any great things I'm going to sell you on, but I think I want to sell you on the concept that we really need to start thinking differently about our minerals. And I think we've started to see that happen, certainly with selenium. A lot of the work that Don Mahan has done, we certainly realize that sources are different. And I want to look at some new tools that are out there and how we look at these things, and then talk about how we sort of interpret those, because I think that's equally important. I'd like to acknowledge my co-author, Blair Aldridge. She's really the one that did all the work. She was a PhD student in my lab. She's currently doing a postdoc at Washington Medical School in St. Louis. And I'd like to acknowledge Alltech, too, for supplying the funding and for this, as well as some of the technical help on the microarray work that we did. So I always like to start with a few quotes or disclaimers. I think we've all sort of, thought this probably as we went through our graduate training, new graduate students, you will by the end. Sort of realize that the more you know, the more you realize you don't know. Change that a little and say, really, the more information and data I collect on any one thing, the more confused I get. Okay, and I think we've seen that today in a few areas where we know we need to advance the science more. We're trying, but the more information we get, you're like, man, I really thought I had that figured out. Crap, now I got to go feed my sows 2 different diets. Okay, so there's lots of those things that, as we evolve, these things change. And so just an example of that, and I've used this before, so hopefully not too many people have seen it. But when we look about minerals, of all minerals, we really should know calcium, right? If you look in the literature, there's tons of work on calcium. Okay, human side, animal side. If you go to any textbook, it gets you some great figure like this. And we absolutely knew that there's a protein called TRPV6 that was responsible for the active absorption of calcium into the cell. We knew that once it was in the cell, it was bound by calbindin, and that transported it across the cell. We knew that with certainty. We knew there was nothing else, 100% certain of it, till some guru had to go in a mouse and knock this protein out, as well as the transport protein. The mouse should have been dead. Mice looked pretty normal. Okay, this was 5 or 6 years ago. Okay, so even something like calcium, the whole community got pretty much stood on their head and said, these are important, obviously, but there's something else going on. So I sort of put that up front as a caveat. We're talking about copper. Try to go to literature and look for a whole lot of data on copper in pigs. Right? We've had some work. Gretchen Hill's done some great work. But if you really go and try to dig for a lot of details, it's a handful of studies, most of them relatively old. So I think we've realized for years that a mineral is not a mineral. We know that phosphorus is a great example of that. We know we can't just look at total phosphorus content. We've got to look at how available that is. I think in my simplistic thinking for years, I thought, well, that's really important in terms of how the mineral gets absorbed. But once it's absorbed, once we have phosphorus or once we have copper in the body, once we have selenium in the body, my thought process for a while is, well, once it gets in, it should act pretty much the same. Okay? And the reality is they don't. Okay, and so when we start to look at different mineral sources, it's not as simplistic as how bioavailable those sources are. It's how they act once they're absorbed, how they're handled by the body, what genes they may or may not affect. Okay, and so it's a lot more information. It muddies the water. Okay, because I can pick one gene that'll make one source look great, and another gene that'll make another source look great if I tell the right story. Okay, so we have to look at the total picture and be careful not to sort of get lost in the weeds. So with minerals, the biggest thing in the last decade or so has been looking at sort of inorganic versus organic. These terms drive me absolutely insane, simply because you can't have an organic mineral. So what we're really referring to, as everyone knows, is not the mineral itself, but what that mineral is bound to or complex with or chelated to. Okay, and whether that is organic or not. And so, with copper, we've historically fed copper sulfate. We now have other forms of copper out there, including some organic forms. You can go to the literature. There's certainly plenty of studies that look at the bioavailability of these. You can believe which numbers you want. And a lot of the sort of organic versus inorganic story has really focused on, what happens to that mineral in the gut. And so if we bind a mineral or complex it to a different substrate, we change the chemistry. And so we change the pH at which point that mineral dissociates from its carrier. And so we affect where in the gut that mineral becomes free. And our sort of assumption for a while was this dissociation has to occur, and at that point, that's when the mineral can be absorbed. Okay, and so if we affect where that happens in the gut, how readily that happens, we can affect bioavailability. More recently, there's been work looking at the mucus layer and saying, well, maybe with some of these organic minerals, that complex actually stays intact longer than we think, and it actually sort of hands the mineral off through the mucus layer to a protein receptor that's involved in absorption. And so that's kind of been the focus. These are small, but this is just looking at, we know that once absorbed, we can have impacts on certain genes. There's hormonal control of a lot of these active transport proteins that are involved in mineral absorption. So in terms of copper and some of the major players, and I'm not going to put up a lot of detail, just try to not put up a lot of chemistry this late in the day. But we know that with dietary copper, we've got the cupric and cuprous form. We know that there's two major, well, we think we know, that there's two major transport proteins involved. One is sort of a generic divalent metal transporter, which will transport other minerals as well. And then there's a transporter known as CTR1 that's believed to be involved in copper transport in a monovalent form. Some more recent work out of our lab that Blair did, actually in her master's, was really looking at a copper peptide and whether that could actually be absorbed intact. And this is one of those things that we believe can happen. I'm not going to even try to speculate what percent of copper can be absorbed in that form. My guess is practically this is a fairly low number. Okay. But I don't know that there's any way to prove for sure that this happens. We have been able to, in a using chamber system, challenge with copper, measure the copper still bound to the peptide in the tissue, and even on the other side. We also went in and we used something that should block this peptide transport. When we did that with copper sulfate, we essentially saw no effect or maybe even a slight increase in the amount of copper that was in the tissue. Whereas when we did it with a copper triglycinate, we saw a fairly large decrease. Okay? And so at this point, we felt fairly confident that this absorption can occur. And so our thought was at that point, well, what's the next step? And so we sort of came up with a hypothesis that if we feed these copper in different forms, okay, and if there is some absorption that occurs through a different route, okay, copper, when it's normally absorbed, has to be bound by a chaperone protein fairly quickly. And our thought was, well, if it's being absorbed still bound to a peptide, it's not an immediate threat to the cell. So likely we would see some differences in gene regulation. So maybe we would see a downregulation in some of these chaperone proteins that normally bind copper. And so that was our thought process. So then we kind of said, well, which genes are we going to pick? Okay, and there's a host of copper chaperone proteins that have been identified. My worry anytime you go on any of those lists is, are you picking the right one? Okay, so we opted to try to go down the microarray route, which we'll talk a little bit about, and really look at as many genes as we could. So our objective was really to determine if organic and inorganic copper differentially regulate genes in the intestine. And so I'm not going to do a lot of details on the methods here, but we basically took newly weaned pigs. We put them on just a standard commercial starter for 3 days, just to get them up and going. We then moved them to a diet that had no added copper. It's not to say it didn't have any copper, but the only copper would have been in our grain sources. We do have analysis on that, so we added 0. We did that for 4 days in a pen feeding situation. We then moved them to individually pens, continued them on that diet for an additional 3 days. Okay, so after a week of this no added copper diet, we then started our 7 dietary treatments, which I'll show you on the next slide. They were on those treatments for 14 days. At that point, we euthanized the pigs and we collected samples. So our dietary treatments, we had 0, 4, 25, or 125 parts per million of copper. Our rationale there was we wanted a true negative control. 4 parts per million is near the requirement. Normally, you would think this is a little low. Really depends if you're calculating. Normally, we ignore the mineral content of our grains. So if we do the calculated, we're a little under 8 parts per million. 25 parts per million, which would be closer to where most of the industry is feeding. And then we wanted to get close to pharmacological dose, and so we went with 125 parts per million. Again, that's probably half of what normally people are going to feed if they're feeding copper sulfate for short periods of time at elevated levels. But we wanted a high level. Okay, and you can see our calculated content of what we should have had if we were adding in what's in our ingredients. Analyzed, we've got some differences, but we were relatively close to where we expected to be on those diets. We then, at the end of the study, we collected jejunal tissue, which we did the microarray analysis on, and we collected all of these tissues with the possibility of doing that. At that point in time, it was about $1,000 a sample, so we limited what we were going to do, and we just compared the 0 and the 25 of each of our copper sources to just see, do we have anything to go further? We then conducted what we're calling an ionomic analysis on the jejunum, kidney sample, liver sample, the contents of the gallbladder that we collected at slaughter, as well as a serum sample. And the idea behind the ionomic, you heard Ryan talk about metabolomic. We all know about genomic. There's lipidomic. I often call it fundingomics. I mean, if you can get that in your grant. And so the idea, though, is that really, let's look at as many minerals as possible, right? And so historically, it's easy just to look at the mineral and assume you're only affecting the mineral that you're looking at. The reality is we know there's tons of interactions, okay? And so this is the idea, is to get as complete a picture as you can. So all this is done by ICP mass spec for those. And we did that on all the tissues listed here. So the idea behind the microarray, we use the porcine Affymetrix chip, and the idea is really to get, I got another omic for you, is really to look at the transcriptome. And so basically we're looking at genes that are being actively expressed, okay? And the technology is moving fast. You go to the mouse world, they're getting rid of this altogether and actually just sequencing the whole transcriptome. But the idea is, to really look at all of the genes and how you're affecting patterns of genes, not just individual genes. So, in a nutshell, there was 214 genes that were regulated differently when we compared 25 parts per million copper sulfate versus copper perrhenate. Okay. So, we've got an effect. This is kind of what I thought. Oh, great, I see these all the time from basic science talks. Well, we affected a lot of genes. Okay, well, that's nice. I'm not sure what it means. Okay. And this is where I think that the agriculture sector is close, but we're way behind the mouse people, and I think in another 10 years, we're going to have a lot better tools. Because the power of a lot of these tools of microarrays and fully sequencing the transcriptome isn't so much the technology to do that, it's the bioinformatics tools on the back end that let you make sense out of all that data. There's 26,000 potential transcripts on that chip, and you don't even want me showing a slide of all 216 and explaining what each gene does and what my interpretations are. So the power of the bioinformatics tools is they let you start to look at these genes and say, okay, are there any genes, is there a pattern here? do we have a pathway where maybe 40% of the genes in a given pathway are regulated, okay, by copper? And if so, that's probably a lot more powerful story than picking out the one gene that tells the story you want it to, okay, because we can always find that. And I'm not going to spend a lot of time going through any of these things in terms of details. But you can see here, this is just looking at basically some different pathways that ultimately regulate gene expression. Okay, and none of these are particularly strong, in my opinion, in terms of the number of genes that are affected. But we can start to see some patterns evolving where we do have a downregulation of certain genes in pathways that are ultimately going to affect gene expression. In this case, they're downregulated by the copper proteinase source versus copper sulfate. And so that's really the power of the bioinformatics tools, and that's where we're heading. I think the challenge today is that we're still really converting all the pig transcripts to mouse gene transcripts, hoping that goes okay, and then we're searching the mouse database essentially, or the human database. And there's some inherent flaws in that. Highlight a couple quick things. We do have, things that maybe get people a little more excited. We do have some potential pathways that affect growth, okay, cell proliferation and differentiation. And so when we start to look at some of these, what I'd call sort of signal transduction molecules, things like protein kinase A and cyclic AMP, we start to look at, okay, we're now affecting genes where practically I can see where this may affect the performance on the ground. Okay? And so we're looking beyond just how bioavailable is the copper. We're looking at what does it do to cell metabolism, what does it do to cell differentiation, what does it potentially ultimately do to growth once it's absorbed. And I think that's where this sort of technology gets very exciting in the future, particularly as we further develop these bioinformatics tools. The flip side of this, instead of looking at what was different between the two sources, is how many were common. Okay? So if we look at our 0 part per million diet versus our 25 part per million diet, there was 474 transcripts that were different as we added 25 parts per million copper versus the control. There was 316 that were different, either up or down regulated, when we added our copper proteinase source. There's only 71 that overlapped. Okay? So we actually had far more that were different between the two sources than in common. So again, copper isn't just copper even once it's absorbed. So we can start to classify these as to whether they're transmembrane receptors. We had quite a few phosphatases and transporters affected. And we can start to put these into categories. What always worries me on these lists that often get left off is over half of them, we don't know what the heck they are. Okay? So we are, because we've submitted some papers and we're submitting some more, we're trying our best to tell the story of what we think this means. I think the challenge is we might be missing the big picture. So I think some big take-home messages from the gene expression part of this, we sort of went in with the hypothesis that if we feed different sources of copper and there's potentially some differences in absorption, we should see some differences in the expression of potentially copper transporters or chaperone proteins. And at least those that are known, there's absolutely no difference between those. Okay, so our assumptions going in, our hypothesis going in is wrong on that level. But we did have 216 that were different between the sources. And if you start to look at the role of copper, we think of it typically as a cofactor in enzymes, but there's a whole host of transcription factors that it appears to regulate. Okay? So copper may have a very large role, expanding role, we know of some of these, in actually regulating various genes that may affect things like growth, the immune response within the body, and that appears that some of those are different between sources. There's also a whole host of genes involved in signal transduction, anything from ion channels to G protein coupled receptors. that were affected by the addition of copper, and there were some differences between the sources. So, sort of to summarize this part, copper source, organic and inorganic dietary copper do alter gene expression, okay, and things involved in cell stress response, proliferation, cytoskeletal remodeling, and signaling. If we look at the 25 parts per million, it does not alter the expression of what we consider traditional copper-related genes. Okay? And there's not one single biological network that I can identify that says, here's where the big impact of copper is, here's where the big difference between sources is. Instead, it has a rather slight involvement in metabolic processes, such as the signal transduction that I indicated, phosphorylation, and stress-responsive pathways. As to the absolute impact of that, I'm not going to try to guess. It's there. I think the next step is to try to hopefully put some numbers to what this potential impact is and does it actually mean anything. So the second part of this experiment was really looking at tissue mineral concentrations through that ionomic profiling. And I'm going to start fairly simplistic. I'm just going to look at copper. And the details here aren't necessarily important, but we're looking at intestine, liver, kidney, and the gallbladder contents here. And just in general, what you'll see across all of these is there's either between 0, 4 to 25, there's either no increase or very minimal increase as we go from 0 to 25. It's really at that 125 part per million level where we start to see large increases in tissue concentrations of copper. And so to me, what that means is that somewhere between 25 and 125, we're really exceeding what the body can handle from a homeostatic mechanism in terms of copper. And we're really starting to exceed what it can handle, so it's accumulating within the tissue. The gallbladder is working very hard to get rid of that copper, but obviously, we're still getting large tissue pools. If we look at the serum, story is a little bit different. This looks like this has a little more tighter regulation, which probably isn't a huge surprise. We do have a copper concentration by source interaction here. Where at our lower levels of copper, we see higher concentrations of copper in the serum if we're feeding copper sulfate. When we go up to 125, that reverses. And so that's sort of the simplistic story, just looking at copper. Most of us have probably seen some sort of mineral wheel like this and prayed it wasn't going to be asked on an exam. But we know and acknowledge that these mineral actions exist. And then for the most part, when we go to formulate our diets, we try to ignore them because we'd never be able to finish formulating our diets. And we try to consider all these. Okay? So we know that these exist. We know that there's things that are involved. I mean, calcium and phosphorus are the classic story. Calcium's positively charged, phosphate's negatively charged. We know that those two will bind, precipitate out a solution within the gut. And as a result, we can decrease the absorption. Okay? To the extent that we use calcium carbonate or limestone as a mineral source in agriculture, and if you go to the human medical side, they call it a drug and use it as an oral phosphate binder. Okay? So it's a very effective phosphate binder. And so we know that we can precipitate. We know in some cases that minerals share the same transport system, and so they can competitively inhibit each other. Usually this is a result of having similar chemical structures. We know that a lot of our minerals, copper for sure, is a cofactor in many enzymes, and some of these will regulate the release of other metals or minerals. We know that in some cases there are shared transcription factors, so if we upregulate transcription factor based on a mineral deficiency, it may affect more than one mineral. And so the idea behind getting this full ionomic profile is we really looked at, I think, 18 or 19 different minerals and tried to get a feel, is there any pattern that we can observe between copper sources across different levels? And I'm not going to show most of that data. I'm going to show you just a little here and then give you sort of the take-home bullet points. And so this is all intestinal tissue. We're looking at molybdenum, magnesium, iron, and zinc. And we're looking across copper sources. Okay? So for this particular set of data, there's no interactions between sources and concentrations. So it's been pooled within each level. And so, with molybdenum, magnesium, we see linear decreases. Okay, most of that is really being driven by that 125 part per million level, whereas with iron and zinc, we actually see increases in intestinal content. So this is the tissue of those minerals. And so just to kind of give you the highlights of what we saw. So if we look at intestinal tissue, we saw a linear increase in copper, iron, zinc, and cadmium. We saw decreases in magnesium and molybdenate as we increase copper level. In the liver, we saw linear increases in copper, selenium, lead, and cadmium. In the kidney, we saw linear increases in copper, iron, selenium, cobalt, cadmium, and manganese. Before I go there, and I think this is where we have to be careful. And my grad student was guilty of this. So her big conclusion initially from this was, and this may be real, but I think we have to be careful. Oh, we increase cadmium. We're increasing heavy metals when we feed high levels of copper. Okay. And that's where you have to be careful and go back and look at the actual numbers. Because as technology advances, the sensitivity on the ICP mass spec, I think, is in the parts per trillion. And so, some of these, yeah, it's increasing. It was pretty consistent. But I think we need to think about, what does that actually mean physiologically? And is this anything we need to be concerned about at the concentrations we measured? I seriously doubt it. But you put in the wrong place that copper supplementation increases heavy metals, could be a problem. If we look in the serum, copper source altered, not as many. Okay, so we only had two things there, molybdenum as well as selenium, that were affected in the serum. And in both of these cases, the copper proteinate resulted in higher concentrations of those minerals in the serum as compared to the copper sulfate. And really the only interactions we saw were a source and concentration effect was in the gallbladder contents, okay, where we had interactions on a number of different metals. Okay. And this may not be surprising, in terms of the gallbladder is trying to excrete most of these minerals. And so it may be a little less surprising here. The other thing we have to consider, we have gone back and looked at this, is you have to look at the purity of your mineral sources because there are contaminants in some of these. We don't think that's what's driving this, but it's something that has to be considered. So I think some take-home messages. Copper source and concentration affect gene expression. That's not rocket science. I think the thing that's really intriguing to me is that there really is a source effect. And it's pretty large. And so, that thinking, well, once we get the copper absorbed, copper is copper, it's not so much the case. Okay, we do have some big effects in terms of what genes are expressed, what genes are upregulated and downregulated. There are huge differences between sources. There's a few that we're working on now that we think are potentially pretty exciting. But I think, we're still a long way from saying, we know these differences are here. I think we're still a long way from saying what's the commercial impact of those. But as we start to look at different mineral sources and as we start to move forward in the future, it's going to be important to realize that just putting it in the diet and seeing how the pig grows, that's ultimately got to be done. But we've got to think about what else we may be doing, what we may be doing to the immune system of that animal. And interestingly, it's not necessarily genes I would have predicted based on what we thought. And then, just looking at the overall copper data, somewhere between 25 and 125 parts per million, I think we're really surpassing the pig's ability to sort of homeostatically regulate copper. And so I think we're, to some extent, overwhelming that system. And so I think if we looked at gene transcription at a much higher level, we probably see a very different set of genes that are affected. So with that, I'll take questions if we have time.